The following is a selected video from masterthecontent.com, where you will find an extensive video library of lectures for a variety of standardized admission tests. We offer over 600 hours of detailed video lectures for a multitude of standardized tests. Use our interactive in-lecture table of contents to find specific topics of interest. Work through numerous in-lecture examples to help you internalize concepts. To learn more, visit masterthecontent.com. Your career, our passion.
I'm going to hop into the first slide of our lecture, which basically introduces these four levels of structure. Now, it's important obviously to define what a protein is to begin with. So it's a biomolecule. It consists of one or more polypeptide chains. You can also think of polypeptide chains as polyamide chains. So basically what that refers to is just a long chain or a long string of amino acids that have been bound together. So the monomeric units or the monomeric subunits in a polypeptide chain would be amino acids, the individual subunits that comprise the chain. So, like I said, amino acids are obviously the simplest, the most basic units of structure in a protein molecule. And as they bind to one another by means of covalent bonds, they can form long polypeptide chains. Now, there are four main levels of protein structure, obviously. So there's the primary level of structure, which I already briefly talked about. It refers to the precise or the exact sequence, the order of amino acids in a polypeptide chain. The secondary structure, on the other hand, refers to the patterns that are formed as a result of hydrogen bonding in the backbone of the polypeptide chain. And I'll explain what I mean by that. So we're basically talking about hydrogen bonding between atoms that form what is the backbone of a polypeptide chain to form regular repeating structures such as alpha helices and beta pleated sheets. Now that constitutes what we call the secondary level of organization. Now, the tertiary level of organization, like I said, is basically what happens when the protein actually starts to fold or conform in a three-dimensional way. So the tertiary level of structure basically refers to the geometry of a protein, and it's actually caused by interactions of the amino acid side chains. So if you remember your basic amino acid structure, we just covered those lectures. Amino acids tend to have what is called a side chain, and the side chains actually allow or permit the tertiary level organization to occur because of the interactions that happen between them. So I'll direct your attention to the middle of the screen right there. We have pretty much what is a segment of a polypeptide chain, a protein. So the exact sequence of amino acids in this chain, the exact sequence of these amino acids is what we call the primary level of structure. Now the secondary structure is actually formed by hydrogen bonding. Now remember what a hydrogen bond is. It's basically a bond that's formed between electronegative element and a hydrogen atom, which is also bonded to another electronegative element. Now in the case of an amino acid chain or polypeptide chain, we have such bonds present. As you can see, the amino group right here has a hydrogen bound to it. Now nitrogen is an electronegative molecule or an electronegative atom. And we also have this carbonyl group adjacently located right here, which has those electrons that it can use to hydrogen bond. So hydrogen bonds can essentially form between the electrons of that oxygen of the carbonyl group and the hydrogen that is held by that nitrogen atom. So the regular repeating structures, or the structures that are resulting from the accumulation of these bonds, are what we call alpha helices and beta pleated sheets, and we're going to study those at length in coming slides. So that's what results in the secondary level of organization. Now at the tertiary level, the entire protein, the long polypeptide chain, starts to form or conform or fold into a three-dimensional shape, and that's caused by what are the side chains. Okay, so as you can see here, R1, R2, R3, those would be the side chains in that polypeptide chain. So they can interact, resulting in what we call the tertiary level of structural organization. Now, at the quaternary level, we're basically talking about more than one polypeptide chain coming together to form one complete functional protein. So that pretty much sums up, or gives you a very basic introduction as far as the 4 levels of protein structure are concerned. Now, you might be somewhat confused at this point in time, but trust me, we're going to go over each and every one of these levels of structure at length in the coming slides, and any questions that you have, I'm sure, will be answered. So before we actually do that, we are going to talk about the versatility of proteins in general.